Welcome back to the Answer Series Diagnostic Report Teaching and Learning Tool for the NSC November examination. This is an all-in-one tool that combines the November NSC paper, Memoranda, and annual Diagnostic Report commentary to create a comprehensive learning experience. TAS provides this document for free, and we encourage teachers, learners, tutors, and subject advisors to use it. All we ask is that you please give credit where credit is due and not change or alter any content in this document that removes our name from it. Question 3.1 talks about genetically modified maize. The bacterium Bacillus thuringiensis produces a toxin called the Bt that kills insects. This bacterium is used to genetically modify maize to contain this Bt toxin, and then the Bt maize becomes toxic to insects. So question 3.1.1 asks to describe how the Bt maize is genetically modified to be insect resistant. Now, candidates lacked a fundamental understanding of the basic steps involved in genetic modification. They gave the steps involved in recombinant DNA technology, so they were explaining how to produce insulin, for example, instead of explaining how genetically modified organisms are produced and applying the process of that to the given example with Bt toxin produced by maize. So let's understand this before we look at the memorandum. We've added some revision in this regard. So below is the basic process of genetic modification. This is extracted from our The Answer Series Grade 12 Life Sciences Part 2, page 75. And a mutant gene is replaced with a desired gene in this example. So this is not always the case. It's not always a case of trying to correct something that is defective within an organism. Sometimes scientists merely want to insert an additional gene or a gene with beneficial characteristics, like in question 3.1 with the Bt toxin being inserted into maize plants. So to answer a question like that, 3.1.1, you have to adapt this basic process to fit the given examples. So you're going to replace the highlighted sections with the specifics for that question. So if we just look at the basic process first, we have a donor cell. In this case, it's a healthy donor cell because, again, this example is where there's some other defective gene. So this donor cell could now be the bacterium, and it contains a desired gene. There's some other gene inside the cell that needs to be transferred into a different organism so that organism becomes genetically modified. Now, this desired gene is isolated or extracted using enzymes, and then it can be transferred into the recipient cell in many different ways. So in this example, there's a viral vector. It's often the way that this is done is they insert the desired gene into a viral vector that then has the capacity to infect the recipient cell and integrate that desired gene into the recipient cell's genome, into that organism's genome. So in the example here, there's a mutant gene that needs to be replaced with a healthy desired gene so that the recombinant DNA is created, and this genetically modified cell, every time it multiplies now, it has this corrected desired gene in it, and that can produce the correct protein. So now if we take this basic process and we apply it to the example in question 3.1. So the desired gene in this example is going to be the Bt toxin gene. That is the characteristic that we want the maize plants to be able to exhibit. So the table here shows the 6 facts that you need to supply to explain any genetic modification process, and you're going to see how this is adapted to question 3.1.1. So the donor cell is the one that is the cell that is the source of the desired gene. And in this case, it's going to be Bacillus thuringiensis. That is the bacterium that contains the Bt toxin and that is capable of producing it. The recipient organism or the recipient cell in this case is going to be maize plants. Specifically, it will start with their cells, and as those cells then grow into plants, the whole maize plant will then have the ability to produce Bt toxin. The desired characteristic, what we would like the plant to be able to do, is to be insect resistant. So how is the recipient cell, how will the maize cells be modified? They will be modified to produce the Bt toxin by inserting that gene into their genome. And the effect of this is that the maize plants will be insect resistant because they now produce the Bt toxin. So let's see how to adapt this general picture here so that it fits within question 3.1.1. So this table shows you all the things that you need to supply when trying to answer the typical genetic modification steps, how to explain how that happens. We need to tell the examiner what is the desired gene, who is the donor cell, where is the gene coming from, who receives this, what cell or organism receives the desired gene, what is the characteristic that this gene allows the organism now to exhibit, how is the recipient cell modified, and what is the effect of this, what is the result or the product that is going to occur. So the picture we saw on the previous page can now be adapted so that it fits the question 3.1 in this NSC paper. And I have done it in two different ways, because you have the visual representation of the picture here where the specific labels that need to be changed have got their colors, and then we also have a paragraph statement which would be typical of the answer. So if we look at the picture, instead of having a healthy donor cell, we now have the name of the donor cell. This is Bacillus thuringiensis, and inside it there is the Bt toxin gene. That is the donor cell. That is where the gene is coming from. So that Bt toxin gene is isolated, and then if you wanted to, even though the memo in this regard did not give any marks to how exactly the gene was transferred into the recipient cell, we have a viral vector step where that isolated desired Bt toxin gene is inserted into a viral vector, and this viral vector transfers it into the maize cells. So the maize cell will now receive the Bt toxin gene, and it will be integrated into the maize plant's genome. So now you have a genetically modified maize cell that contains a Bt toxin gene, and therefore that cell is now capable of producing the Bt toxin protein, which then means that the plant as a whole becomes insect resistant. If we look at the paragraph, that's exactly how it's been worded. So the Bt toxin gene extracted from Bacillus thuringiensis' DNA, transferred to the cells of the maize plant, that's the recipient, via a viral vector. The viral vector then inserts the Bt gene into the DNA of the maize plant cells, and those plant cells can now produce the Bt toxin. So therefore the cells and the plant, or the organism as a whole, becomes insect resistant. So this is a nice table to kind of summarize and to use for practice with different examples of genetically modified organisms. So let's return back to the question. So back on question 3.1.1's memo, we see that any three of the following marks were awarded. The Bt producing gene was cut from the bacterial DNA. That was inserted into the maize's DNA, so they don't give a mark for any way of how that insertion happens. That recombinant DNA then causes the maize plant to be able to produce the Bt toxin. So the example that we work through is a much more complete answer and a much more complete description of the process, whereas in the memo, any three of the following could have been provided. Question 3.1.2 says to explain 2 reasons why farmers might want to grow Bt maize. So Bacillus thuringiensis being able to kill insects and now transferring that ability to plants means that the insect that would normally eat a farmer's crop will die. So they won't have as many losses in terms of their crop. The crop will therefore provide a higher yield, and that higher yield means that the farmer will receive more profit, more income, more money. They could also have said something about increased food security, or that the crops would be healthier in general, etc. A different perspective would be that if the crops are producing their own internal toxins that would kill insects that normally eat them, a farmer wouldn't need to spray as many pesticides. Therefore, the farmer is actually going to save money by not having to buy those expensive pesticides. It also means that the crops would not contain as many toxins and be healthier for consumers as well as the environment. Then question 3.2 was concerned with human evolution. It shows a phylogenetic tree that shows the evolutionary relationships between some species. The first question asks to give the letter representing the common ancestor for all the species shown in the phylogenetic tree. So that requires you to go all the way back down to the root of the tree, which shows that there was a common ancestor V, and all the lines branch from that point upwards. So V is the common ancestor for all the species shown in the phylogenetic tree. Question 2 asks you to identify the species that is most closely related to Old World monkeys. Now, in order to answer this question, candidates had to remember that species that share the most recent common ancestor are also the most closely related. So you would put your finger on Old World monkeys, and then you would trace that line back to where there is a common ancestor. And if you go down, the first common ancestor that appears is you. And then from you, the branch that goes up is the closest branch in terms of distance is gibbons. So gibbons are the most closely related species to Old World monkeys. So it says in the diagnostic report that many candidates could not interpret the phylogenetic tree, and they said that orangutans are the most closely related to Old World monkeys just because they are right next to one another. But if you look at how the lines work, you would go all the way down to U, then up to another common ancestor T, and then from there they only share a connection to orangutans. So gibbons are actually closer related because they share a more recent common ancestor at U. So candidates did not trace the lines of origin back to gibbons and instead chose the species that were indicated next to Old World monkeys on the diagram. The marking guideline also provided a mark for U as an option, as candidates were not necessarily incorrect in saying that U is actually a species in and of itself. It's some other common ancestor that is obviously the most closely related to Old World monkeys. Question 3.2.3 asks you to give a reason for your answer in question 3.2.2. The reason here depended on whether learners said gibbons was their answer or U, the common ancestor. So if learners said gibbon, then the answer here would be that they share the most recent common ancestor at point U, U representing the common ancestor. If learners provided U as the answer in 3.2.2, then the reason for their answer would be that Old World monkeys directly descended from U. That is their most recent common ancestor. Then continuing with this question, 3.2.4 asks us to identify 3 species that share the common ancestor R. So you would go to the diagram, you would put your finger on the R, and you would look at in which direction R branches. So directly it branches to gorillas, then it goes up to Q, where there's another common ancestor, but from there it moves on to humans, and it also moves into chimpanzees. So those are 3 species that have actually been identified in the diagram: humans, chimpanzees, and gorillas. The marking guideline also provided a mark for Q, which represents one of the common ancestors. It's some other species that we're not sure what it was, so it gave learners the benefit of the doubt there. Then question 325 says, state 3 characteristics of the upper limbs that humans share with group P. So group P, your orangutans, gorillas, and chimpanzees are all closely related relatives, and they are all larger great apes, basically, that have the similarities that they learn about between humans and great apes in terms of the evolution section. So there were quite a number of similarities that they could have chosen. So freely rotating upper arms, long upper arms, that there's rotation around the elbow joints, rotation around the wrist, bare fingertips, that they have nails instead of claws, that they have fingerprints, that there's an opposable thumb or a precision grip of some kind, and that all their limbs have 5 digits, or we say that they have a pentadactyl hand, meaning 5 digits on the hand. So any 3 and only the first 3 were marked. Incorrect responses by candidates in this regard included saying that they had long arms instead of long upper arms, writing that they have 5 fingers instead of 5 fingers per limb, writing that they have nails instead of specifying that they have nails instead of claws. So it's not the full phrase or the full idea that's being communicated here. That they were referring to other body parts, like, for example, the skull or the feet, etc., when the question specifically asked for differences in the upper limbs, and then also discussing the significance of bipedalism, which had nothing to do with the characteristics of the upper limbs that these organisms share. Then question 3.3 is the age-old scientific investigation, and the content here involved evolution and genetics. So a group of researchers conducted an investigation, and now we have the aim that we need to underline, to determine the influence of the type of milk, that is our independent variable, on the height of the children, that is the dependent variable who drink the milk. So our aim is already identifying for us the two variables that we are very likely to be asked in some sense. They said that the investigation was conducted over a period of 7 years from the age 3 to the age of 10 years. In terms of their setup, they selected 4,146 healthy 3-year-old children of the same race and the same gender. They divided them into 2 equal groups. The one group was given a 1 liter of cow's milk to drink daily, and the other group was given 1 liter of soy milk to drink daily. They ensured that each group had the same additional diet, the same daily activities, and the same living conditions, because obviously those things would have affected their growth as well, and we want to control for those variables. And they provided the milk from the same supplier, and then they measured the children's height at the end of the investigation, so at the age of 10 years, and they calculated an average. So the table shows the results at the end of the investigation compared to the expected average height of a 10-year-old, probably based on just general data. And what we can see is that the expected height of a child of 10 years should be 149.86 centimeters. The average height of children on cow's milk was above that at the end of 10 years, whereas the average height of children on soy milk was below that. So the first question asks us to identify the independent variable and the dependent variable. We've already done that by just looking at the aim of the investigation. So we highlighted that the type of milk is the thing that is being manipulated by the investigators. So I determine the type of milk. I, the investigator, manipulate that. So that's the independent variable. And what is that affecting? It's affecting the height of the child. And that's also what we're measuring. So that's my dependent variable. The diagnostic report says that candidates wrote the influence of the type of milk. In our paper 1 video for this year's diagnostic report, a similar problem occurred with the question on auxins, talking about the effect of the auxins. So here we have influence. The influence of the milk is actually what we are measuring. So learners incorrectly wrote influence of type of milk under the independent variable. They literally copied and pasted it from the aim, and this is where they went wrong, because the influence of the milk is actually what we're measuring. That is the dependent variable. That is going to affect, it's basically determining the height. So what exactly are we manipulating is what type of milk we are providing. So please remember that influence is actually the height. So that's the dependent variable. Some candidates incorrectly took the headings of the table and then used those to describe the variables. So now we can see in both papers of this year, there was a problem with the writing of the independent variable, with words being phrased into the aim that learners then just copy and paste, and then they lose marks because they're actually providing the dependent variable. Question 3.3.2 asks to name the type of variation displayed in the height of a person, and this is pure learning work, part of the section on evolution where learners are taught 2 types of variation, continuous and discontinuous. Continuous variation is where we have a whole spectrum of phenotypes from one extreme to the other. We don't have distinct categories, like we don't just have everyone is either 149 meters long or 0, right? Height is over a spectrum from very short people to very tall people, so it's an example of continuous variation. Then the next question asks learners to state 2 planning steps that the researchers had to consider at the beginning of this investigation. So this is a nice question in the sense that the memorandum gave a lot of possible options for answers. And what we like to tell learners at TAS about writing down planning steps is to always think about who, when, and where. Planning steps will very seldomly count a lot of marks, so you need to have at least 3 that you can have in your little pocket to pull out. So answering who means determining who your target group is, determining the size of your sample. When means, what date am I going to do this? What time frame is connected to it? Where refers to the venue. Where is the location of this investigation going to take place? And then another one that always appears on marking guidelines is asking for consent, especially when you're working with kids or with adults, is actually getting people's permission. Other examples of things that learners could have provided includes ensuring that the same additional diet was provided, ensuring that they had daily activities that were similar. So that would have required a lot of planning beforehand, ensuring that the living conditions were quite similar, to train research assistants, to decide how you're going to record your data, designing recording sheets, deciding on, what sources of milk are you going to make use of, what apparatus or equipment will you be making use of, and choosing the type of milk and also the amount of milk. So candidates did not differentiate between planning steps and procedural steps. And that's something we highlight in our scientific skills section at the beginning of our books, which says that planning involves decisions that are made before the investigation, whereas the whole procedure, the conducting, involves decisions that are made after planning, and it's actually part of the investigation method. So many candidates just copied the first bullets of the procedure as part of their responses. So remember, for planning, who, when, where, and ask for permission. If you can give at least those 4, you will have enough planning steps. Then they ask for 3 ways in which the validity of the investigation was ensured, which means you need to go back to the description of the investigation and go and highlight everything that was kept the same. Remember that validity has to do with variables, VV. An investigation is only valid if it tests what it was intending to test. There are no other things that could have resulted in the same outcome. So we need to control for all variables that would make our investigation less valid. So we read, they had children of the same race, the same gender. They had 2 equal groups. So the number of children in each group was the same. They gave them the same amount of milk, 1 liter of soy milk, 1 liter of cow milk in each group. Each of the groups had the same additional diet on a daily basis, similar daily activities, similar living conditions or environmental conditions. The supplier of the milk was the same, and they only used healthy children, which they mention right at the start. And then another one is that the duration of the investigation was the same. So they conducted it over a 7-year period. So both groups were subject to this investigation for a total of 7 years. So candidates did not grasp the concept of evaluating the validity of an investigation. Then question 5 asks learners to state one factor that could have negatively affected the validity of this investigation. So obviously, besides all the things that they've now mentioned, the investigation actually controlled for, there was this whole list of things that they did do. You need to now think of anything that they couldn't control or that they didn't mention as controlling that could have possibly affected the height of the children. And one of the most important things is the fact that even though they chose, healthy 3-year-old children, all of us have a completely different genetic makeup. Some of us might just have genes that allow us to grow taller, regardless of the fact that we might be ingesting soy milk. So the genetic factors were not considered, were not controlled for, or even the hormonal influences as they go into their teens. It's difficult also to really maintain the same conditions. I mean, they're talking about similar daily activities, but not exactly the same. Similar living conditions, but not exactly the same. So controlling those might definitely have posed a big problem and affected the validity of the investigation. And then there's no measuring of a baseline height that you could actually maybe work out a ratio of how particular kids grew compared to what their height was initially. Candidates did not grasp the concept of evaluating the validity of an investigation. This is the same comment as for 3.3.4, which also involved validity. So validity really remains an issue for learners to understand. And again, we've provided a number of other resources that help teachers to teach the different concepts within scientific investigations. Question 6, how did the researchers ensure the reliability of the results? So remember that reliability has to do with repeating the investigation, increasing the sample size, random sampling, and readings, taking more readings and calculating an average. Now, the problem with this is that we've drilled those generic responses into learners' minds for so many years that now they just keep giving the generic responses like increase the sample size instead of applying it to the context of the investigation. So we need to tell them, great, if you can list those things to me about what makes an investigation reliable, can you tell me which of those they actually did? So if we look at the memorandum, they got a mark for saying that there was a large sample size of 4,146 children that participated. That's obviously a lot more than having 2 children participating, which means the results are more reliable. Then you could also have commented on the number of kids in each group, because the groups were also quite big. They divided them equally, which meant that there was about 2,073 children in each group that participated. So again, that is a large sample size. So those would be your two most important reasons for how this investigation is basically considered to be reliable. The memorandum also includes the fact that you have quite a long duration, so a 7-year span over which the investigation was conducted. Now, this is not a typical response in exams, but maybe the argument here was that a longer time frame reduces the impact of random fluctuations or errors that might have been introduced into the data if the time frame was really short. So it allows for a more stable and a consistent measurement of whatever phenomenon is being studied, which leads to more trustworthy results at the end of the day, more reliable results. Then the last question is to state a conclusion for the investigation. So as we mentioned right at the beginning, if we look at the results in the table, we can see that the average height of a child who was drinking cow's milk for 7 years is higher than the expected average height, and that of the soy milk is slightly below average. So in order to formulate a successful conclusion, you have to link the results to the aim of the investigation. So the aim was to determine the influence of the type of milk on the height of children drinking the milk. So we have to basically say, what did we find? What did we find in terms of which type of milk affected and influenced the height of the children in what particular way? So there are many ways to phrase this. Examples include cow's milk leads to better growth or increased height compared to that of soy milk. Soy milk consumption by children leads to a decreased height or a below average height, or cow's milk consumption by children leads to an increased height above average height. So many candidates incorrectly provided an analysis of the results. So they said something like the average height of children who drank cow's milk was 2.46 centimeter taller than those who drank soy milk. Children who drank cow's milk had an average height compared to those who drank soy milk, which was X or Y. So depending on how the results are presented, an analysis of the results may sometimes provide the conclusion, but this is not always the case. So candidates have to read and comprehend the context of each investigation and not apply generalized rules. Then question 3.4 on human evolution says there's a passage about Homo erectus fossils. Evidence from fossils of Homo erectus shows that they appeared in Africa about 2 million years ago. They spread to many parts of Asia. Homo erectus is the oldest known species to be fully bipedal and have a human-like body. Their brain size was smaller than that of humans today, and a prominent brow ridge was present over their eye sockets. Question 341 asks learners to describe how the fossils of Homo erectus are used to support the out of Africa hypothesis. So this is exactly from the exam guideline that the oldest fossils of Homo erectus were found in Africa, while younger fossils were found in other parts of the world, for example Asia, that it's mentioned there in the paragraph, which indicates that Homo erectus originated in Africa but then migrated out. Question 342 asks learners to describe how all other fossil evidence is used to support the out of Africa hypothesis. And again, this is referenced directly from the exam guideline, where learners basically just have to comment on what fossils that are older than Homo erectus have only been found in Africa, and what fossils that are younger than Homo erectus have been found outside of Africa. So if you think about the sequence, we first had Ardipithecus. Fossils of Ardipithecus have only ever been found in Africa. Australopithecus was the next common ancestor, and the fossils of Australopithecus have also only been found in Africa. Then we have Homo habilis, which is the first fossil species for the Homo genus. Fossils of Homo habilis have only ever been found in Africa so far. The oldest fossils of Homo sapiens have only been found in Africa, but younger fossils of Homo sapiens have been found in other parts of the world. So that basically creates this picture that in terms of our ancestors, our common ancestors before the Homo genus, have only ever been found in Africa. And if they are our common ancestors, it means that life basically in terms of human nature originated in Africa. Then in terms of Homo habilis, the first species of Homo, the Homo genus, they have only been found in Africa. And if they are our ancestor and only been found in Africa, it means that we originated in Africa. And then going up to Homo erectus, which has already been answered in question 341, Homo erectus has moved out of Africa, so we do find fossils of Homo erectus in the rest of the world. But the important thing is that the oldest fossils have only been found in Africa. Younger fossils are found in the rest of the world. And the same pattern repeats with Homo sapiens. Homo sapien fossils have been found in all parts of the world, including Africa, but the important thing is that the oldest versions of these fossils have only been found in Africa and younger ones in the rest of the world. So candidates did not correctly describe the role of fossils in supporting the out of Africa hypothesis, and key words, for example, oldest or only been found here, were omitted or incorrectly used. The question continues to ask learners to use the information in the passage regarding Homo erectus and to tabulate two differences between the features of Homo erectus and Homo sapiens. Now, when a question asks to tabulate, it means that if the question counts 5 marks, one of those marks will be for actually just drawing a table. So even if any learners are listening, if there is nothing you can fill into the table, at least just draw a table and write the two things you need to compare with one another, and you would get 1 out of the 5 marks. Now, in terms of what is mentioned in the paragraph, there were only these two differences. The brain size was mentioned, that Homo erectus has a small brain, so Homo sapiens obviously have a larger brain, and they also mentioned the brow ridges, which is prominent in Homo erectus, and in Homo sapiens we know that they are reduced brow ridges. So not large and small. Please note the wording here, prominent brow ridges compared to reduced brow ridges. Candidates listed differences between Homo erectus and Homo sapiens outside the information that was provided in the passage. They also did not align the differences per row in the table, so they were actually putting them cross from one another. So they listed brain size in the first column and then brow ridges in the second column, instead of comparing the brain size of Homo erectus to the brain size of Homo sapiens and the brow ridges to the brow ridges of Homo sapiens. Question 344, what observation of the skull of Homo erectus would have led scientists to conclude that Homo erectus was bipedal? So this has to do with the position of the foramen magnum. That is the only indication on a skull that could indicate bipedalism, which is that the foramen magnum is in a more forward position. Not central, but more forward. That is the phrasing that they prefer in examinations. So candidates incorrectly describe the position of the foramen magnum as being in the middle or in the center of the skull, which has been mentioned in previous diagnostic reports as being incorrect. Then question 3.5 was on evolution through natural selection. In 1986, there was a huge radioactive disaster in Chernobyl, and all humans immediately left the area because of the deadly high radiation. Animals exposed to this high radiation developed cancer or died, and almost 40 years later, scientists have been studying the wolves that live in this highly radioactive environment where they are exposed to high radiation, and they found that these wolves have a mutation that makes them immune to cancer. The first question asks learners for a definition of a mutation, basically. What is a mutation? So mutation, as defined in the curriculum, is any change in the genetic composition of an organism. The second question, what effect does exposure to high radiation have on animals? If we read the paragraph, it says animals exposed to this high radiation developed cancer or died. You could also have said that it causes mutations, as that is the whole theme of the article that has been provided as an extract. Then learners were asked to use Darwin's theory of natural selection to explain the development of immunity to cancer in these wolves. So in the latest version of our Life Sciences Part 2 class text and study guide, we've provided worked examples of how to structure answers for Darwin's theory, Lamarck's theory, speciation, etc., which really helps learners to structure their answers effectively. So when answering Darwin's theory on natural selection, you always start by asking yourself, what are the different characteristics? In other words, you describe the variation. So you say, there was variation amongst a population of wolves. Some of them had a mutation that made them immune to cancer, and others did not. Okay. Then you ask yourself, what changed? Then something happened in the environment. So there was a selection pressure. They were exposed to radiation. How are the different organisms now going to be affected by this? Describe who had a favorable trait and who had an unfavorable trait. So the wolves without this immunity died. The wolves with this mutation or immunity survive. And this is basically what natural selection is. So then you have to think, okay, describe the inheritance, what inheritance of this favorable trait occurs. So the wolves with the mutation that survived would reproduce, and they would pass on this allele for the mutation or for the immunity to their offspring, which means that the next generation of wolves would have a higher proportion of wolves with this mutation or immunity to cancer. And we also revised this whole process of natural selection answers in a previous diagnostic report that can be downloaded from our website. Candidates lost marks because they gave a generic response and they didn't apply this to the natural selection example. They used the terms favorable and unfavorable characteristics without actually saying, in terms of this example, what is favorable and what is unfavorable. So having the mutation was actually favorable in this case because it made them immune to cancer. Not having the mutation was unfavorable because those wolves were susceptible to cancer. They could not identify the characteristic in the wolves that was the source of variation, or they incorrectly identified the variation as a resistance to radiation instead of an immunity to cancer. Candidates also referred to evolution of species instead of the evolution that occurs within a population of wolves. They incorrectly stated that the characteristic of immunity to cancer was passed on when it's actually the allele. So again, confusing the word characteristic, which is the phenotypic expression, and allele, which is the genotypic combination. Incorrectly, they stated that the wolf population will now have the characteristic of immunity when it's actually the next generation of wolves that have a higher proportion of individuals with that favorable immunity to cancer. So very common misconceptions and errors here, and this would be a great question to work through with learners in class and to actually tell them, okay, who of you did the same thing wrong that they did in 2024? So then finally, suggestions for Section B's Question 3. The following issues were reported in the Diagnostic Report of 2023, but they continue to be a challenge, and you will note that we have provided revision in this regard in a previous Diagnostic Report teaching and learning tool. The knowledge that natural selection occurs in populations, not in individuals and not in species. That characteristics in natural selection are favorable and not desirable. Favorable is in natural selection. Desirable has to do with genetic modification, where we can decide, I desire that characteristic and I'm going to put it into the genome of a different organism. And then also providing generic responses and questions on validity, reliability in scientific investigations. In terms of scientific investigations, again, not to provide generic responses. Remember to teach learners that they need to apply those generic responses to the specific procedure or data that has been provided. Typical questions that are asked in scientific investigations that we need to prepare learners for is to identify the variables, that is the independent, the dependent, and the control variables, to be able to define what a control is and also why is it needed, to express the need for a control, to state the aim of an investigation, to list planning steps for an investigation, to suggest how the investigation may improve its validity or even just how it did ensure validity, to suggest how the investigation may improve its reliability or just how did it ensure that it was reliable, to state a conclusion and to evaluate the procedure. In other words, what could they have done better? What did they do wrong? Now, TAS has provided extensive revision and resources on the scientific method besides the diagnostic report and teaching tools referenced above. If you click here, you can see all the offerings that we have available. So on YouTube, we have a webinar that we did on scientific method and that you can watch. There are also resources that go with that. There are short videos to explain all the individual components of a scientific method, like the aim, how to identify variables, how to understand validity, reliability, etc. There are downloadable web page resources. So in reference to the webinar above, there is the basic handout of the scientific method procedure that's available in both English and Afrikaans for download. There's also a handout on how to read and interpret a scientific investigation question, and we've also provided a practice test and memo in both English and Afrikaans that can be used. The previous diagnostic report and teaching tools have got examples of questions that learners struggled with in terms of scientific investigations, and we've provided some teaching tools and tips in that regard. And then in our study guides themselves, there's a whole skills section in which we not only go through the scientific method, but we have a worked example of a scientific investigation and also the type of questions that could be asked, as well as memo answers. And there are scientific investigations in most, if not all, of the topic questions in our study guides. So have a look at those. Continuing on to the next page, that also mentions that the successful interpretation of phylogenetic trees can only be mastered through practice. And the following features of phylogenetic trees needs to be emphasized. The species that occur within the present time, so where is the present time, where is the past? The point of extinction of a particular species, species that exist during the same time period, the difference between a common ancestor and the most recent common ancestor, and species that have a more recent common ancestor are more closely related. And in our newly revised Grade 12 Life Sciences Part 2 class text and study guide, we have a number of useful single-page summaries, and one of them is shown here in a little sneak peek on phylogenetic trees that can really assist learners in understanding how to read and interpret these diagrams. Evolution, especially human evolution, is taught towards the end of the academic year, and therefore it may be glossed over. This explains the lower performance of learners within these questions. So ensure that there's sufficient time and effort placed into the delivery of this content. And then lastly, genetic modification, which was the question that was most poorly answered in this paper, is a problem. So use basic steps to assist learners' understanding of this process, and we've included examples of this on slide 50 of this document to help you practice. So that brings us to the end of this year's teaching and learning tool for the paper 1 and paper 2 of life sciences. I hope you've learned something new and that you can convey this to your learners.
If you found this video useful, give it a like and subscribe to our channel so you don't miss any new episodes. Follow us on Instagram or Facebook to stay on top of the latest TAS news and launches. So that's it for now from the Answer Series, your key to exam success.